So far in our development unit, we've focused on the process of growth. Now we're going to turn our attention to the process of differentiation. Let's start just by reviewing development. Development is the transformation of a single fertilized egg, a zygote, into a multicellular adult. And this involves two steps. The first one is growth. The zygote divides by mitosis to make identical cells. It also involves differentiation, where those cells acquire special shapes and functions. There are many different cells in your body. Here are 3 different types of cells. Each of these different cells has different structures and different functions. In other words, different traits. However, each of these different cells came from the same zygote. Came from the zygote that divided by mitosis to produce genetically identical cells. So even though these cells have different traits, they all have the same DNA. If you were to look in the muscle cell and the pancreas cell and the blood cell, you would see the identical DNA sequence. So this is a difficult paradox to explain. How can cells with different traits have the same DNA? Well, to understand, we need to quickly review how genes determine traits. Remember that genes, or DNA, are the instructions for building a cell's proteins. And this is done through the process of protein synthesis, when DNA is transcribed into RNA, which is then translated into protein. And your proteins are ultimately your traits. So, going back to the question at hand, gene regulation explains how cells can have the same DNA but different traits. This is a microarray, which shows what the genes in a white blood cell are doing compared to the genes in a pancreatic cell. Note that both of these cells came from the same individual. What's different about these cells is that they're using different genes. All the genes here that are dark blue are always turned on. In other words, they're being transcribed and translated to produce a protein. The genes that are red are turned off all of the time. They are never transcribed or translated into a protein. And meanwhile, the light blue and the yellow genes are sometimes on and sometimes off. So what's different about a white blood cell, or a pancreatic cell, or a bone cell, or a brain cell, is the genes that it is using to produce a protein. And this is gene regulation, turning on or off transcription and translation of genes. And another way of saying that a gene is turned on is to say that it is being expressed, being used to build a protein. And in any particular cell, some genes are never expressed, never turned on. Some are always on, and some are on some of the time. This process has a lot of advantages. If you consider a pancreas cell versus a white blood cell, one advantage is that it allows cells to specialize in certain jobs. The pancreas can be really good at just producing the protein insulin, whereas the white blood cell, a B cell, can be really good at producing the protein antibodies. It also helps cells conserve resources. The pancreas cell isn't going to make every single protein possible when really its job is just to make the insulin protein. Same thing with the white blood cell. This also lets cells respond to changes in the environment. For example, the pancreas cells only need to produce insulin if blood sugar is too high. If blood sugar is normal or too low, it's a waste of time and energy to make insulin because insulin isn't needed. Similarly, if there's no virus or bacteria in a person's body, there's no need for a B cell to go to the trouble of making antibody proteins. It's a waste. So how do genes get regulated? How are they turned on and off? Well, this is a simplified mechanism for how it's done. Basically, there's a signaling molecule that's released from one cell that binds to a receptor protein in the membrane of another cell. And when it binds, it sends a signal to another molecule, which signals another molecule, which signals another molecule, which signals another molecule, which finally signals the DNA to be transcribed into mRNA, which can then be translated at the ribosome into a protein. So long story short, signal molecules will tell a cell to either start or stop transcribing its genes. So let's zoom out a little bit and go back to embryonic development. Gene regulation is closely related to the process of embryonic induction. And this is the process where a ball of cells will become a specialized fetus. So let's zoom in on these 2 or 3 cells. What's happening with these 2 or 3 cells is that one cell is sending a signal to another cell. The cell that sends the signal is the inducing cell. The cell that receives the signal is the responding cell. When the responding cell gets the signal, genes will be turned on and transcribed and translated into proteins, and other genes might be turned off. This will cause the cell to specialize. It will get a specific structure and function. It then might send signals to another cell and tell that cell to turn on and off certain genes, and that cell will then become specialized. So it's kind of like a domino effect, but the domino effect is precisely controlled by one cell sending signals to another cell. And that is embryonic induction. So let's just summarize everything we've learned today. During development, a cell can differentiate into specialized cells by activating different genes. So this cell is activating genes that help it look like a neuron, and this cell is activating genes that help it look like an epithelial cell. And as a result of this, cells differentiate and acquire special structures and functions.